All right. So, lab 5, enzymes. Hopefully, you've gotten through the chapter 5 content on enzymes, which will help you understand this material much better. So, when we look at this, we're going to be looking at catalase. Catalase is an enzyme we find in potatoes. I would have literally, if we were in the classroom, taken a potato, put it in a blender with some water, blended it up, and strained it, and we would have gotten this soupy mixture that we would be using as our catalase. When we mix catalase with hydrogen peroxide, it breaks down. This is a catabolic reaction, and we end up getting water and oxygen, and literally this forms bubbles. It's kind of a fun lab to do. So our reactants would be the hydrogen peroxide. The products would be water and oxygen, which forms bubbles. And notice that our enzyme is over the arrow. Okay, so because it is over the arrow, it means that it is not part of the reactants or the products. So when we look at enzymes, we have, and this is kind of our vocab here, we have our substrate. So in this case, it is hydrogen peroxide. It is going to fit in that active site. When it fits into the active site, it forms that enzyme substrate complex. And in this case, we're going to break that hydrogen peroxide down into water and oxygen. So the first lab that we are going to do is we're going to see which of the substances. We're going to use 4 different substances. We're going to use water, hydrogen peroxide, glucose, and protein. So you would have 4 test tubes. You would have those reactants in them, and then we would have added the catalase and measured how high the bubble column would be. That bubble height would indicate whether the catalase is working or not. It will not work on molecules that are not its substrate. So what you're going to do next is you're going to pause this video and then go to the 5.1 video where we have Marcia Hay is going to do this experiment for you. So go ahead and watch it. And then once you get done watching it, we're going to go to the next slide, and that's where I'm going to give you the numbers. All right, so pause it now, watch the video. All right, now assuming that you watched the video, these are the numbers I want you to put in your lab book. She didn't give you no numbers. She just kind of gave you, she showed you the test tubes, but she didn't measure them. So here are the measurements I'm giving you. We do sometimes see a little bit of bubbles just because of mixing the stuff up, but that doesn't mean necessarily it's the products we're looking for. So you can clearly see here that the substrate for hydrogen peroxide or for catalase is hydrogen peroxide because it clearly produced a lot of bubbles. The next lab that we're going to do is to see which temperature the catalase works best in. So again, you would have a test tube in ice water, in an incubator that's kind of around room temperature, and then boiling water. So what I want you to do next is again, pause this video and watch that procedure. Here are the numbers that I want you to use for that particular lab. What we find is we generally find that the highest one is in the ice water, but we generally get some pretty good, a significant amount of bubbles with the incubator. What I would suspect is that probably the ideal temperature for potato catalase is in between those 2 temperatures. But most groups will get the ice water making the most bubbles. The next lab we would have done is on pH. Which pH does this enzyme work the best at? Does it work best at an acid, a base, or neutral? So again, Marsha's going to show you that lab. Please pause and watch that procedure. All right, now I'm hopeful that you did in fact watch that. And then we have our results. This is another case where usually like half of my class will get the neutral being the highest for them, and then the other half will be the base. And again, I kind of suspect the ideal temperature is probably in between 7 and 11. The next lab is probably one of the harder ones to understand, and that is where we're looking at enzyme concentration. So we're going to look at the amount of enzyme. So I'm going to think of the enzyme being the painter. So notice I have two rooms. They have the same amount of wall space. We are going to have the same amount of paint. So let's just say that we had the same size bucket, right? Same size bucket in both of the rooms. Now, again, our bucket of paint would be our reactants. Okay. So I'm asking you how much our product would be so our product would be our painted wall. Painted wall. So we have the same amount of reactants. We have the same amount of wall space. But our workers, and that's what your enzymes are. Your enzymes are your workers. In this particular lab, you're going to have different amounts of enzyme. And so if I asked you, all right, if I told these painters, I said, you have 10 minutes to paint the room. On your mark, get set, go. And then when their 10 minutes are up, I say, stop. Who would have the most wall space painted? Well, ideally, it would be the one with the more painters, right? The 3 painters, because they have more enzymes or more workers. When we get done, though, if I said you had unlimited time, how much product would room 1 have versus room 2? The product would be the same. They have the same amount of paint, they have the same amount of wall space, they're going to have the same results. It just will take this poor guy here a lot longer to obtain those results because he's up by himself. So there's less workers. So that is the concept we're trying to get you to understand with this particular lab. All right. So here is our concentration. So again, what I would like you to do is pause. So we're looking at, let me just go over this real quick. We are looking at 1 centimeter, 2 centimeters, or 3 centimeters. So kind of think of it as 1 painter, 2 painters, or 3 painters. So again, pause and watch the procedure. Here are the numbers I would like you to use for this lab. So again, we gave you 20 seconds and we said at the end of 20 seconds, measure those bubble height. Well, the one worker only got 12 millimeters of product. 2 centimeters got 23 and 3 got 35. And then the last lab actually combines a few labs together. So in this case, we're not looking at catalase, we're looking at amylase. Amylase is an enzyme we find in your spit, in your saliva, and it breaks down carbohydrates. It takes these complex carbohydrates, right, our polysaccharides, and it breaks it down into monosaccharides, our poly to mono, which is why when you eat things like crackers, they're not very sweet to begin with, but then once you chew it up into a paste, it gets sweeter and sweeter and sweeter. Now, starch, which is our polysaccharide here, is not a reducing sugar. And we did that. If you look back in your reducing sugar test, starch did not give you a positive result. Glucose is a reducing sugar. So this is a reducing sugar here. This one is not. Okay. So what you're going to do is you're going to go ahead and you're going to do a couple different tests. You're going to test whether it's a reducing sugar. That would be the Benedict's test. You're also going to do a starch test, right? Because starch will test positive for starch. Your monosaccharide or your glucose is not going to test positive for a starch test. And so you're going to do a bunch of tests on these different test tubes. You need to watch the video and fill in the results she's going to show you, because you're looking at color changes. So you will get your information from that. If not, it is also, I have it in the lab review. So again, watch the video, then do your virtual lab. You're going to want to, because as you filled out your lab book, you're going to want to check your lab book with the key, right? I always have the key there so you can make sure you filled it out properly so you're not studying the wrong information. And then you're going to go ahead and do your lab review, submit it, and then take your quiz. As always, you have any questions, please send me an email. All right, good luck.